Hey team, welcome to another screencast. Today we're going to be talking about membrane transport. And this is an important thing to talk about because remember, a cell, essentially a bubble of fat, is in an environment where there are lots of good things potentially on the outside of the cell that need to come in, and lots of, or potentially some bad things on the inside of a cell that need to go out, and also potentially bad things on the outside of a cell that we don't want to come in. So how the cell controls the movement of molecules through its membrane is very important. So we're going to be talking about membrane transport. And there's going to be a lot of terminology coming at you, so watch out, but I'm going to lay it out pretty carefully, so I think you will get it by the end. Okay, I'm going to scroll down here and start fresh. Okay, so there are two basic types of transport. And the first one is passive. Oh no, I don't want to write in red. Okay, passive transport. And then there is active. And the difference between the two of these is passive does not require energy. So no energy needed. And active requires energy. Okay, and passive transport can be broken down into two different types of transport. We have simple diffusion, and protein-mediated passive transport. Okay, so those are the two types of passive transports. I'm going to come back to those in a second. And then we have active transport, and again we have two different types of active transport. Active transport, much like passive, we have protein-mediated active transport, as well as bulk transport. Okay, so there are the two different types of categories and broken down into their constituent pieces. Okay, so let's get a little bit more in depth here. So we said passive transport doesn't require any energy. And so you should know that this type of transport is driven forward by molecular motion. We've talked about this before. So the second law of thermodynamics tells us that all things are trending towards thermodynamic equilibrium, and so they're moving down their concentration gradient. So passive transport is always from high to low. And like we said before, it's driven forward. This movement from high to low is driven forward by molecular motion. So let's create a cell membrane really quickly here. I'm going to take some time to draw this in. I'm going to pause this first. Okay, so here we've got our phospholipid bilayer, and we know that we've got this fluid mosaic model. And the fluid part of this model tells us that these particles are constantly moving around. All of them, not just this one, but this one as well. Ooh, that's not spinning around, but we know that they're moving up and down and around. This is what the fluid part of this model tells us. So there are constantly teeny tiny little pores opening up through this membrane, and this allows the diffusion of small uncharged molecules. For an example, CO2 is a small uncharged molecule, and oxygen, O2, molecular oxygen, that's another small uncharged molecule. These things can move through the phospholipid bilayer down their concentration gradient. So for example, if the concentration gradient of oxygen is high outside the cell, it's going to be moving into the cell. And if the concentration gradient of CO2 here is high inside the cell, then it is going to be moving out. And that's going to happen just through simple diffusion, because these pores, these micropores in the cell membrane, are opening up all the time. So there you have it. That's simple diffusion, driven forward by molecular motion, always from high to low, and it's passive. It requires no energy. So I'm going to write that down. Simple diffusion, we have small, uncharged molecules moving from high to low. And that could be either from the inside to the outside or the outside to the inside. Okay, now let's talk about protein-mediated passive transport. Now we have the same cell membrane right here, and let's imagine we have a larger or charged molecule, and it can't move through the membrane itself. Now, I'm not going to tell you why it can't move through the membrane, but I want you to remember something, that this membrane is hydrophilic here and hydrophobic here. And you are going to have a class quiz, a group quiz, over the next couple days, and I'm going to be asking you questions about movement through membranes. And so I'm not exactly going to tell you why large charged particles can't move through the membrane. You're going to have to figure that out over the course of the next few days. But I want you to know for this screencast that they can't. So to move large and charged proteins through the membrane, we have proteins embedded within, oh, this is a bad drawing, but imagine we've got this protein and it's a pore, and they're often gated pores, so they're not open all the time. So imagine, I'm going to grab some of this membrane again, put it on the other side. Okay, imagine we have this protein channel embedded within our membrane. This is why this is called the fluid mosaic model. Not only are these things moving around in the membrane, there's also mosaic pieces. There are different pieces embedded within the membrane, and part of them can be proteins. We also talked about carbohydrates studied on the membrane. But remember the fluid mosaic model. Okay, so protein-mediated passive transport is the same type of thing, except now, if you can imagine, we can move larger charged molecules through these membranes. Before, we were moving small molecules, so for example, molecular oxygen was little, and CO2 was also quite small. Ooh, where did everything go? Oh, I accidentally flipped onto a new page. But CO2 is a small molecule, so they can move through the membrane. These protein channels allow for larger charged molecules to go through, but this is still moving from high to low, and things are going to, again, move down their concentration gradient. So if there's a large concentration out here, then things are going to move through the channel this way, into the cell. If there's a large concentration in here, things are going to move through the channel out of the cell. So again, passive mediated transport is small, charged, still high to low. Sorry, did I say small? I meant large. Large. Okay, great. Okay, let's move on to active transport. So as we said before, passive transport requires no energy because it's relying on molecular motion. However, active transport requires energy, and this energy is in the form of ATP. I think we've talked about ATP before briefly. ATP, oh, not ATB. ATP is the energy currency of the cell. It's like, for example, you can only use dollars in America, but if you go to England, you have to use pounds. Well, ATP, if you go to a cell, that's the energy of the currency of the cell. That's how We can spend energy in the cell. And active transport requires ATP to drive forward. And this is because in, for example, protein-mediated active transport, we've still got a protein embedded within a membrane like so, but in this case, we're moving large charged or small uncharged against the concentration gradient. So the big key here is with protein-mediated active transport, we're moving from low concentration to high concentration. So we can't rely on molecular motion to drive this reaction forward. We've got to spend some ATP to move things into the cell. But this can move large charged or small and uncharged. I don't like the way this is grouped. Okay, so basically this is things that are moving against their concentration gradient. And the thing to note here is that things that are moving through the membrane by simple diffusion, small uncharged molecules, we can't really keep them in a place against their concentration gradient because they just move through the membrane easily by themselves. So basically, protein-mediated active transport, only charged things or large things or small things that are charged, we can only control their movement through the membrane, because small uncharged things, they're just going to go through. We can't do much about it. Okay, and the last type of transport is bulk transport, and there are two types of bulk transport that we're going to talk about. The first one is exocytosis. This is things leaving the cell. And then we have endocytosis. This is things coming into the cell. And there are two types of endocytosis. There's pinocytosis. And this is taking in small drinks. Pino is, I think that means small in Latin. But it's small, taking in small vesicles. And then phagocytosis, which is large things. Okay, and both these things require energy, not necessarily because they're moving from low to high concentration like protein-mediated active transport, but because they require vesicles. And the fusion, both the creation of fusion of vesicles with the cell membrane requires molecular motors as well as a whole array of proteins that are going to be responsible. They're called V snares and T snares. It's more depth than you have to know, but they're going to be responsible for binding to the cell membrane. So this type of transport requires a whole lot of energy because of the movement and the motion of the vesicles. It's basically like moving a semi-truck full of goods around in the cell. It requires a lot of energy. Okay, so this is it. Passive and active transport. And again, this is all important because the cell has got to maintain homeostasis. Okay, that is it, folks. Have a good night.